Hello, Dr. Wong. It's nice to have you today. My name is Tamara Ekis, and I'm the president of the Reflections of Grace Foundation and also the vice president of the IPG DMG collaborative. And we are so proud to have you here today to talk a little bit about your grant. But before we do that, we would like to present you with a check for 100,000 US dollars for your grant titled Exploiting Nuclear Vulnerabilities in Pediatric High-Grade Gliomas.
Thank you for that. I didn't expect this coming so quickly today. I thought we were just having some discussion on the grant applications, but I really appreciate the support uh, that you have given to us because over the years we have been very drawn to study DMG. I'm sure you know about Rob DeRose. We started this journey into this research probably about 10 years ago when he shared with us the story of the passing of his daughter. And that's how we got into this research. Yeah.
Well, that's wonderful, and we're so happy to have you, and we're so happy to start off with the great news that we have for you today. I'd like to hand it off to Martin, who would like to say a few words.
Thank you.
Hello, Dr. Wong. I'm also very happy and congratulate you to this fantastic support. I'm just now sitting, not standing, on the absolutely other side of the world. I'm from Europe, from Prague. I represent both the IPG-DMG collaborative and also I'm the representative of Mediation Foundation. We are very happy that you exploit absolutely different region of interest in possible future management of this very severe disease. And let me to congratulate you. And I'm very happy that I can now introduce. I don't see Jackie, but yeah, okay. So that's why I don't see you. So let me introduce Jackie, who would like also to give you a few words.
Thank you.
Thank you, Martin. I am here. I hope you can see me.
Now, yes.
But Dr. Wan, thank you very much for joining us. Thank you for the work that you do. My name is Jackie Criswell, and I'm with the Cure Starts Now Foundation. But more importantly, I hold the title of proud aunt to Elena, who is a DIPG angel. Families like us, this is what makes your work so much more important, and it means that much more to us. So thank you for what you do. Congratulations on your grant. Very much look forward to seeing what can come of this.
Yeah, we're looking forward to work with you as well on this.
Thank
Yeah.
you. And I think Keith would like to say a few words as well.
Hello, Dr. Wong. I'm Keith Desrick. I'm with the Cure Starts Now. I founded the Cure Starts Now, and I also helped found the DIPG DMG collaborative. And I know we've had some correspondence back and forth. And it's nice to finally award this grant. It's nice to kick off some of the questions we may have about the grant. But first, tell you that you won. I think that kind of helps.
Thank you.
I know this is also not only just an international with Martin and many of us around there, but you have a lot of Australian partners as well that are really, really proud of the work that you're doing and really want to pass on their congratulations. I know you referenced the Isabella Marcus Foundation. They're also a member of the collaborative. Also, the Cure Starts Now there in Australia, run DIPG. Robert Connor Dawes, and Cure Brain Cancer.
Yeah.
They all come together, and this is really a worldwide effort. So congratulations. We also wanted to go through some questions, but I'll turn it over to Rachel so that way she can go ahead and offer her congratulations and learn a little bit more about the grant.
Yep. Thank you.
Hello, Dr. Wong. My name is Rachel, and I'm the Research Support Coordinator at the Cure Starts Now. Congratulations on your grant. We're very excited for you and excited to see the results. One question for you. Your research focuses on hidden weaknesses in pediatric brain tumors. Can you explain what this means in simple terms?
So I guess this has to come back to the problem that we try to tackle to understand the impact, which is with DMG in particular, the mutations of the DNA, it doesn't just change gene activity, but because of the importance of the histone genes, which are frequently mutated, it actually affects bulk of the genome, which has what we call the repetitive DNA. One of the key regions is the ribosomal DNA repeats, which are really important sites for making transcript to make ribosomes, so we are able to produce protein. And in cancer cells, they are highly dependent of effective way of making proteins. And because of a histone mutation, it actually affects these sites for making ribosome. Therefore, a number of years ago, we found that ATRX mutation actually causes repeats DNA instability. And now we see this in the histone mutation, which is frequently found in DMG. They have the similar weaknesses in terms of making ribosomal transcript. And one key things that we discover also is the factor that bring the RNA polymerase to these repeats are UBF1 and 2 factors. We found that the change in the chromatin due to the histone mutation actually affect this UBF1 binding. And we have now discovered more of this even in the patient tissues as well, working with Royal Children's Hospital here from their biobanking. And we are really excited to explore more about how UBF1 and 2 actually recruited to the ribosomal DNA, because this is the key to bring RNA polymerase to drive the transcript. And we want to do this using obviously chromatin profiling, using imagings, and also doing more genetic screen in order to find other pathway, which we believe will be chromatin pathway. They are also important for making UBF1 able to bind. And with this, together with the PMR116, which is a drug that our collaborators have developed, and has been shown to be effective in pancreatic cancer cell model and also in mouse model. And also in blood cancers, we're excited to bring these drugs. And also hopefully through the genetic screen, we can find more targets. We are able to better target the DMG. And this is also because there are also many other chromatin mutation in brain cancers, in ATRT or other cancers, in IDH1 cancers, for example, in the brains. And we want to be able to not just target DMG, although that is our primary focus. We believe this, whatever we discover here in DMG, will be applicable for other cancers with chromatin mutation. And that's our aim, basically, to establish this hypothesis and to test them, and also to in the futures to bring this into the clinic.